Okay. So, thanks again to Professor Chandra for a very nice talk. We now have the next speaker. Rukmini Kumar from Attage Research.
Hi, thank you again to all the organizers for having this lovely conference. I think the size, the atmosphere, everything, you've hit it very well. So great job on that. And I'm excited to talk a little bit about what I work on. So I work at Vantage Research, and we work as a modeling and simulation CRO that works with drug developers. And I'll talk a little bit about one case study that we have worked for almost like 6 or 7 years now, where we've slowly built evidence to have a great impact, and I think an important impact. So I'll talk a little bit about that, and then maybe a little bit about, a tiny bit about, what is the role of modeling and simulation in drug development today, and what opportunities exist. So that's the agenda today. So let me start a little bit with how drug development really happens today. In an ideal scenario, I think we've heard a lot about we're thinking a little bit more about who's the right patient for what drug. So there's a lot of subtlety involved in omics, and trying to understand what is going on there. Here, I'm trying to figure this out. I see. Now I see. So here we are in a different world. When we're talking about the pharma drug development pipeline, we're really maybe already identified the protein of interest. We've already identified a set of compounds that may attack that protein. We have some accumulated knowledge from biomedical literature, and then we're going to start testing whether this protein is effective in reducing disease burden, first in animal models, and then what we think of as the gold standard for when a drug will come out, which is a randomized controlled trial. And then if the drug is safe and efficacious, better than an existing treatment, that's a drug which is a successful drug and can be administered to everyone. So this is kind of the ideal path of what happens in drug development. However, the reality is much messier. When you start, there are already a lot of uncertainties in understanding the biology. There are a lot of drugs which work in the market today very effectively, but we still don't quite understand what is the exact mechanism. Metformin is a great example of that. It's a safe drug, it's been used for many years, but exactly how it alleviates symptoms of diabetes is still poorly understood. When you start off, there are many alternative biochemical, entities which can go. Which of these should be prioritized? That's often a very important question. Is it the lead? Is it the follow-up? What even is the best marker to track efficacy? So if you think of a disease like diabetes, it's very clear that we want to track blood glucose, and if you reduce blood glucose, you're doing a good job. But even for a very common disease like rheumatoid arthritis, there are all kinds of subjective and objective scales to understand what really is happening in the disease, what should I reduce to alleviate the suffering of the patient. A very important thing in clinical pharmacology is what should the dose be? Should it be the same dose for everybody that walks in through the clinic, or should it be different doses? And how often should we dose? Is it once a week? And these are often balanced with real life considerations. Especially if you want to inject a drug, you don't want the patient to come to the hospital every week. So you want to say, can I push it out to once in 2 weeks? Can I push it out to once in 3 weeks? So these are the various, kind of sampling of the type of questions that we have to deal with. And once, we do cross this threshold, there is a lot of monitoring of what happens in the real world. Is the drug really as safe and efficacious as we thought in the trials? Is that being carried through? At the end of all of this, what is the upshot is drug development is expensive, it's inefficient, it takes a lot of money and a lot of time, and it's a risky venture altogether. So how can modeling and simulation sort of help in this process? That's the goal of what we're doing. I think there's been a lot of, lot of angles to this exact message, which is integrating knowledge, right? When you think of what is happening in a drug, very often your doctor or the MD PhD who's running a trial, they're really thinking about what is happening at the population scale. We're trying to manipulate something. There is variability in the population, but we all understand that what's happening is really several layers deep. And this problem is compounded by the fact that a lot of times this information is siloed. The person who's really designing the drug is a protein engineer. They care very much about what's happening at that scale. There are translational scientists who work with animal models, clinical pharmacologists who really worry about how does a drug interact with the populations. There are MD PhDs. Somewhere in the mix there are modelers. So it's in this complicated landscape that, you know, the big challenges are how do we integrate all this information, and the next big challenge is how do we do it quantitatively. Without modeling, what often happens is, you know, you have a important MD PhD from Yale and he's like, okay, you know, this is what I think the drug is, this is what I think the dose is, and we go with a lot of gut instinct. What you know, we really want to do is to quantify this information so that these sort of decisions can happen objectively at scale. So with that context of what happens in drug development, let me talk a little bit about a case study. This was work done with Merck, Sharp and Dohme, like I said, over the past 6-7 years now. The context, the story with which we entered this collaboration, this was you know, maybe 2016, 2017, and Merck had great success with a drug called pembrolizumab. But great success in cancer still means, you know, 20 to 30% of the patients were being helped by pembrolizumab, and in this case, I'm particularly thinking of melanoma. And the goal was, what is the right combination? What do we want to add to melanoma so that we can improve that survival rate to 40, 50, 60%? What are the drugs which are there in our pipeline that we want to combine with melanoma? Do we want to target these patients differently? Do we want to dose these patients differently? So we came in with a sense of momentum from there, but there were a lot of big questions to answer. So a little bit of a background for checkpoint inhibitors, and I think they really caught the imagination when about 2014 or 2015, the 90-year-old ex-president of the United States, Jimmy Carter, he fell ill with melanoma, and he actually recovered from melanoma, and you know, he remained cancer-free without treatment, and that was kind of a breathtaking achievement which really resonated with everybody, because this would have been a death sentence at most ages for most people, and you know, this kind of caught the imagination of a lot of the general people. Yeah. So what is the magic? What is really happening? How does pembrolizumab work? So the crux of what is happening is, you know, we all understand that the immune system has evolved to really go after anything that is foreign in our body. So a lot of this is basically infections, viruses, bacteria, but the mutated tumor cell is also a foreign antigen, and this in principle can be recognized by the immune system. Now for most of the, you know, from 1950 to 2000, I feel the biggest weapons in our arsenal against cancer was really chemotherapy. And as we all know, that's cytotoxic agents basically designed to poison fast proliferating cells. I think there was always a thread of, you know, the immune system can be, you know, harnessed to attack cancer, and a lot of scientists had been working on it. And you know, the result of that was a checkpoint inhibitor. The way the cancer immune cycle, which is what this famous paper shows, is very similar to the infection immunity cycle, in the sense that the cancer cells release cancer antigen. These are presented to cells of the immune system. The T cells can be trafficked, which are specific to these antigen, via the blood cells back into the tumor tissue, and the T cells themselves can kill the cancer antigen. I mean, the T cells recognize the cancer antigen and hence kill the cancer cells. So this is in brief the cancer immunity cycle. If you drill into this a little bit more, what's happening here is, this is an antigen presenting cell. So this is usually a roving cell which catches all the foreign antigens and comes back and presents it to the immune cells. And what is interesting about the immune system as well is it's always on a knife edge. Too much inflammation is the leading cause of why people die in hospitals. So when somebody dies in the ICU, part of it is the virus that causes damage to your cells, but a big part of it is your immune system overreaction, which causes what we think of as collateral damage to various organs in your body, and that damage can, can lead to reduced energy and reduced organ function and death. So a lot of times when the immune system is being stimulated, there are stimulatory and co-stimulatory signals, but there are also inhibitory signals. So this is a signal where the antigen is being presented, but the system has evolved to always have like the brake and the accelerator next to each other, and that constant balance is what takes you from fighting the infection and bringing you back to homeostatic health. Now, what some tumor cells have done is evolved to evade the immune system by overexpressing these brakes. So this is PD-L1, which is a brake which is evolved to slow down. It is an inhibitory signal which downregulates T cell activation, and that's what's been overexpressed by this tumor cell. Checkpoints merely checkpoints, they remove the brakes. So these are two important checkpoints, and this is how they work. Anti-PD-L1 stops the brake signal, and anti-CTLA4 also stops the brake signal. So let's zoom back up. This is what was happening in kind of the bottommost level. Let's zoom back up to what is happening in the clinic. So when you're a cancer physician running a clinical trial, what are the things that you're looking for? The first thing is what is a classic survival curve. On the y-axis is how many patients can be classified as progression free, which is that cancer is in remission, you're well, your tumors are not growing. If your tumors were growing, you would fall out of this curve. And how many people are we able to keep in remission and for how long? So that concept is called disease progression. What exactly does that mean when a patient comes into the hospital? When a patient comes into the hospital with melanoma, they don't have usually just one single lesion, and these lesions are not all on the skin. These lesions have metastasized to various places. Patients may come up to 5 to 10 to 15 lesions sometimes. So the job of the doctor to track is they pick 2 to 5 of what they call the target lesions. They sum up the diameters and they track those diameters, and that is that's called the sum of longest diameters. And if you look at cancer clinical trials, that's what is being tracked. But the story gets even more complicated, because not only does the patient have the 5 to 10 lesions that they came with, somewhere in the middle of the trial they'll get a new metastasis. Somewhere in the middle of the trial, one of the lesions that you didn't track, that can grow more than you expected. So when you look at cancer trials, you have to think of what is happening to the target lesions, and these lesions doctors make sure they are measurable, they are in easy areas which you can scan. So there are some important characteristics for how to select a target lesion, and over and beyond that you have to keep track of what else is happening in the cancer for the patient. All of these are summarized in a standard score called the RECIST score, and most clinical trials are interpreted in this manner. Now let's zoom back down, because we were talking about the immune system can respond to the tumor. Each patient can have multiple lesions, and these lesions can be of different varieties as well. I think there was one slide to this effect yesterday as well. So when you look at these tumor cells, in a lesion you can have a lucky lesion where there are a lot of immune cells. So the CD8 cells, NK cells, if they are flooding into your lesion, this lesion could be toast, because we'll see, but this lesion at least you have a fighting chance against, because your immune system has tracked this. All the way at the other end are very clever lesions. These have somehow escaped detection, and these are called cold lesions. So there are hot lesions, cold lesions, and obviously this is not a binary scale, so there are intermediate lesions as well. So when you look at that patient who comes in, your standard picture is, okay, there are 5 lesions, they sort of all look the same. Especially in the world of chemotherapy, they may all looked the same and behaved the same under the eyes of the chemotherapy, because they're all multiplying lesions. But when you think of immunotherapy, it's possible that all lesions are susceptible to immunotherapy. All lesions are not susceptible to immunotherapy, but most frustratingly, or what is difficult to interpret, is when there are multiple lesions. So this is the crux of the work that we did with Merck, where we tried to kind of really establish this concept, evaluate it, and figure out what is the impact of this heterogeneity. So here what we have is how a clinical trial is visualized. This is called a spider plot. Basically each line is a patient, and if the patient is really lucky, and the y-axis is how the diameter changes upon treatment. So if you're really lucky, you start, and then within 6 weeks, 90% of your tumor is evaporated. Awesome, you are a responder. Many patients, they linger on the trial, some of them drop out, but really you're able to track them. These are the lucky patients whose tumors or the sum of longest diameters really shrunk by almost 100%. These are the unlucky ones where the sum of longest diameters kept increasing, so your drug really didn't have an effect. So this is a very classic way to visualize what's happening in a clinical trial. As we were digging into the data at Merck, what we realized was very confusing things can happen, and this can really come back to the lesion to lesion variability. So each of those lines there, if you think of it as what's happening to the change in diameter, here's a patient, this is all very polite, you think this is what is happening, but when you imagine it, you may imagine that if this lesion is stable, the diameter didn't change at all, you may imagine some variability like this. But what really happens can depend on the mechanistic, the makeup of that lesion. So if it's a hot lesion, it can shrink, and if it's a cold lesion, it can go out. These are cartoons, and we'll talk a little bit about the data which supports this as well. But before that, let's talk a little bit about why is this important? Why do we care? Is this merely an academic issue, or does this matter? This matters because of the following. We have 3 examples here, A, B, and C. All of them under RECIST criteria would have been classified as progressors. These people did not respond to the drug. They have to drop out of the trial, the drug is useless for them. But on the other hand, if you look at this, each of these patients have very different responses. This patient in blue is literally the least lucky, or they've just not responded to the drug. Every one of their target lesions has grown, all of their non-target lesions have grown, and they've got a new metastatic lesion. So this person genuinely has not responded to the drug that you gave. On the other hand, here's another patient. All of their lesions have shrunk, their non-target lesions have also shrunk, but wait, they developed a new metastasis. Now, according to the old criteria, where again I feel like it's simple to measure, it probably worked pretty well for chemo, this patient is still a non-responder because despite all of this, their new metastasis is keeps them as not under remission. They still have cancer, so this patient is also not a responder. Obviously, there is behaviors in between as well. Now, if you're a doctor, the treatment strategy for each of these could be very, very different. For this patient, you want to take them off the drug that you have, try radiation or surgery or something different. For this patient, you may just want to try radiation at that point, or add another combination, but keep your main drug on because clearly it's helping. So the definitions which kind of push heterogeneity under the rug have real world implications. So that was just a cartoon. We also did analysis to establish this concept. So here is another way to visualize what happens in a cancer trial. This is what happens at the end of that time profile that I showed. So there's no time here. At the end of the trial, what is the best change in diameters that you saw? And this is stacked by patient numbers. So these are all the patients that responded. These are the patients that are somewhere in between. These are the patients that really progressed and your drug didn't help at all. So in the immunotherapy literature, any paper that you look at will have this thing called a waterfall plot. So what we showed was this was data from Merck's melanoma trial. What we showed was when you stack those patients, these are the responders, these are the non-responders. Even among the non-responders, there are lesions which respond completely to your drug. So there is a lot of heterogeneity there. Among the responders, there are still lesions which are growing, which do not respond to the drug. So don't take these patients off the drug. You need to add something else to these patients, or so we have problems in both ends here. And really this is the standard deviation of individual lesion growth and what is kind of being shown as the average in most clinical trials. So heterogeneity is real. It's not just a concept that you can work out from first principles, but this is right here and it has real life consequences. So a lot of this work, it's really great because it started off with modelers and clinical pharmacologists, and slowly we built this framework. And this is a recent publication by our collaborators, and this was a perspective actually. And what they say is the following. They say that there are challenges remain today in how to interpret clinical data, How should a patient with apparent clinical benefit and a subset of tumor sites that are growing and are not growing be managed, or new or growing be managed? Should the checkpoint inhibitor be discontinued, supplemented, orthogonal, site-specific, etc.? And all of these are very valid questions which you can't answer with an open and shut, this person responded and this person didn't. This was published, Walchuk, who's a leader in the field, he and a couple of our collaborators, they published this perspective very recently, and it kind of reiterates the real world implications of this work. So like I said, this is work which started with, and we are definitely not the only group to say that. Several clinical papers exist. If you Google dissociated response or search for dissociated response, there's a lot of work talking about why these criteria have to be changed. But like I said, I think what is really cool and interesting about this work, it was truly interdisciplinary. We started off with modelers, clinical pharmacologists, the MDs within the company, and then MDs outside the company who believed in this, kind of problem that needed to be addressed. So it really took the expertise and energy of a lot of people to come up with this body of work, and it's still ongoing. Right now, we're working on a paper to again show, using the data, that patients that have been relegated as progressors have shown a meaningful benefit from continuing on this original drug. So, work is still in progress. But to kind of change gears and talk to an audience of computational biologists, I think some of you may be modelers, some of you may be looking at the data, and the one thing that I would like to talk about is a framework for considering how you interpret cancer trials. So I think the classic way is you really think of the single prototypical lesion. You have all of these ideas about what is happening inside that lesion, which is great. But remember, and a virtual patient or a synthetic patient in your trial will have multiple target lesions. So each of these target lesions will have different biology, different initial conditions, different growth rates, and that will matter in how you think about treating this patient or simulating this patient. Think about metastasis as a new thing. The way we modeled it is as a probabilistic model attached to a mechanistic model, where we said the more the tumor burden, the higher the possibility of metastasis. There are many other ways in which you may want to do this, but do consider that there are multiple lesions. Metastasis is an almost stochastic event that can happen somewhere in between, and your population of patients has that level of complexity underneath the covers. So this is the key takeaway. Friends don't let friends model or analyze cancer trials without considering lesion to lesion heterogeneity. So do consider that if you are looking at cancer trials. Like I said, there are a lot of people involved in this work. I'm only talking about and all of them are authors in the various publications. But the folks that I worked with a lot are these folks, and Brian Top was really provided a lot of heart and leadership for this effort. Okay, I still have 5 minutes to talk about careers in biosimulation. And so, at the end of this talk, definitely ask me questions about both sections. But what I have noticed is when I come to academic groups to talk about the work that we do at Vantage, there's a healthy curiosity about how does science happen in a non-academic setting, and what do we really do, how do we really function. So I'll just take a couple of minutes to talk about that. The field that a lot of this work happens under, you can think of as quantitative systems pharmacology. It's a complicated name, but it's basically a lot of differential equation modeling to integrate multiple scales. As you can see, the mentions of QSP have really kind of blown up exponentially over the past few years. People who work in this sort of modeling, they can be drug developers and companies, CROs like Vantage, software developers. MATLAB, for instance, has software like this, regulators in the FDA and other agencies, people in healthcare, where you really think of, how does this affect real-time care. Vantage itself, we're a company of about 10 years old now, almost 40 people at this time. And these are the companies that we work with and we have published with. There's another handful of companies that we work with but haven't yet published with. So it's really we are part of the growth story of QSP, because all of these companies need talent and are requiring for this sort of capability. To give you a little sense of how these models are developed, there are biologists, there are modelers, biologists come with baseline degree in immunology, cell biology, etc., molecular biology, etc. Modelers are folks with physics, engineering sort of background, and together we crunch the literature to really understand how to build these models. And sometimes, this is kind of the full-blown how we develop a model. Sometimes the models are semi-mechanistic, minimal, they don't have to be very complicated. Sometimes it's just empirical. Sometimes it's, from literature analysis. But all of this is in service of moving and having more efficient decision making around what we think of as the drug development pipeline, which is discovery, clinical, translation. We're really kind of pushing the agenda with whatever tools it takes according to, along this pipeline. There are several case studies that we have, some of which we're going to start publishing and putting on our website, but that's kind of where we are. So with that, thank you again for the invitation, and we're open to questions now.
Thanks, Rukmini, for bringing us back.
Quantitative systems pharmacology today.
I mean, we do differential equations, partial differential equations.
You need initial parameters and those data points.
Yeah, so that's a very interesting question, and there is a, and I was really interested in Mani's talk because I'm like, hey, can we repurpose your tool to tell us T-cell life cycle rates? Can we use that to kind of figure out, disease prevalences, etc.? So yes, these models require a lot of curation of the literature to really understand how to quantify these various life cycles. So in that model, I showed what is the T-cell trafficking rates, what's the tumor growth rate, what is the cytotoxicity when these two interact. All of those are parameters that we really have to chase down and pin down.
So how do you get them? So that's a curiosity.
Literature survey. So we're not as machine learning like Mani's group. So that's why our group is about 30% biologists and 60% engineers. So this really requires, before building a model like this, and if you look at the citations for any modeling paper, you'll have like dozens of papers which scientists have gone through to really isolate each of these rates and then come back to inform the model.
Some maybe private question. Does industry have more data than we have outside?
Yes, but it is super siloed. It's like, it can even be siloed inside a big company. So the biostats people won't share it with the modeling people who won't share it with some other people. So, data is precious, it's precious everywhere, and we're grateful that we had some access to it. But I actually don't even know the kind of data that I would like, which is disease profiles and data like that, I feel is rare or missing in the public, in public domain. These kind of data, they're not, they're clinical trial data. So it's very, in a very close setting compared to a real world setting. So that's what I'd say.
This final question. Does NDA help here with an academic institution to get the data out?
Say that again. NDA?
NDA, I mean non-disclosure agreements.
Mm-hmm.
Industries like whom you work with, would they be encouraged if to work with academicians if there's a possibility?
That's a very loaded question because, like I said, I think one big difference between academic science and industry science is in industry, any analysis that you do is in service of a decision. So just fishing expeditions, as they call it, they are unlikely to do that with anything which is red hot and going on right now. But old data of drugs which are off patent or old data of drugs which have failed, that's a much more productive area to look for data versus something which is very important to the company's bottom line today.
Thank you.
There are no more questions. Let's thank Rukmini and all the speakers of the day.
Thank you.